Good to see everybody again. Hope you've been well since last week. So today is our second of two sessions. And so if last week, if you were to ask me how to bookend the two parts, if last week was about testing, right? It was really about genetic testing and then making decisions about which of the embryos you tested to implant. This is about modifying. So even 20 years ago, when the story I talked about with you last week happened, it was not possible to do modification, only possible to do testing. But in that time, there has been a rapid revolution of technology which permits, as it says here, editing the genome, not just testing what's in the genome. So, sorry, that's my computer. So that's a ugly screenshot for a reason. Maybe you'll be able to make out at the bottom left there that this is an article from Science dated the 6th of June, 1975. And that's actually a photograph of a hard copy, so it's not a digital version. And it's important because it provides a kind of interesting landmark for the discussion related to governance of emerging technologies. This was about, this is a story reported about an event that took place at a place called the Asilomar Conference Center, which is on the sort of Santa Barbara area coast of California. It's called the Asilomar Conference because of where it was held, but it was important because it was the scientific community getting together to talk about how they would regulate themselves related to the use of a brand new technique that had been developed by the people who are listed here on the authorship line, some of whom became Nobel laureates, to do something called recombinant DNA technology. So the ability to snip, to change the DNA in living things was developed just around this time. So quite a long time ago in some respect, but obviously not that long ago in the scheme of history. And it's important because it was the scientific community realizing that they needed to be responsible for the use of this new technology that was brand new. So it wasn't, it wasn't just emerging, it was brand new. And it had sort of untold potential to both help, but also to do things that might be really perilous. So they talk about things like if we modify a bacteria using these techniques in ways that we didn't intend or didn't expect, and it somehow got released into the environment, what would happen? They were a little bit less concerned about modification of humans because it didn't seem like that was so likely to be immediately on the horizon, but obviously it had the potential to be used that way. So this was the beginning, effectively, of the genome editing debate, even though it wasn't called that, because it didn't have the capacity to edit. It was much more gross in the sense that it was scattershot in the way that it worked, but it was an important point. So published in Science, 1975. That was 20th century, right? So there's some differences between the 20th century and the ability of the scientific community. So important to say one more thing. So they were able to collect pretty much everybody in the world that was able to do this science in one place at the same time. They were all in the US and the UK. Nowhere else in the world was there the ability. And so you could get people in a room and say, how are we going to manage this? And you might have some hope that would actually have some effect. 21st century, very different. Why? So as it says here, science, and you I think saw in the way that the Netflix documentary showed, right, the democratization of science showed up in very clear relief in that that depiction, right, of people who have no formal science training using these tools in ways that nobody's regulating. So there's a good aspect of that. Science is far more democratic, and notice that's small d democratic, not political democratic, than it was in the past. That means there's not one country that can lead, or one, US and UK, two countries that can lead, which makes it difficult to govern. But it is the case that less, expertise, less specialized equipment in laboratories, less training is required. And so there's wider access to tools that can be used in very useful ways, good ways, but also, of course, ways that seem kind of crazy and maybe not so responsible, and of course, without a lot of accountability or responsible oversight. So I made this point, but here it is again. There are just really fewer ways to self-police, fewer mechanisms. You know, we can't have another Asilomar is sort of the upshot of this. And it just can't happen without the concentration of expertise and leadership, which is just not the case now. Okay. So things started to change pretty dramatically when CRISPR and other technologies, you know, CRISPR is the one we know best or hear most about, but they're all tools. TALENs is another one. Zinc finger, ZF, you sometimes see ZFN as the acronym there, zinc finger nuclease. All of these are tools that allow much more precise, careful, targeted changes at the genetic level. And I'm not, for lack of time, not interest, going to, talk about how DNA works, but if you want me to, I certainly can. I don't know if we talked about this last week, but my first degree, my undergraduate degree, was in molecular biology. It was called microbiology. It wasn't called even molecular biology then, but it was very early molecular biology. So I have enough science knowledge to be dangerous. So I can happily explain if you would like. But the tools became much more precise. And there was a realization that we needed to have some kind of an attempt, at least, at getting the international community together to talk about responsible use. And so that's what this next little bit of discussion is about. In December of 2015, so not long after the kind of bursting onto the scene of CRISPR-Cas9 and other techniques as being kind of controllable, and you sort of start to realize the kinds of things that could be done with a technology that had that level of precision, reliability, ability to change make a single change in the DNA of an organism, human, animal, bacteria, plant, anything with DNA could be altered kind of at will. That there needed to be a way for the international community to talk about this. And so, as it says here, this is a news release from the National Academy of Sciences from December 2015. It was an international summit was convened in Washington, D.C., but it was truly international. And there was a collaboration in the hosting of it. It was held in DC at the National Academy of Sciences, but it was the Chinese Academy of Sciences, the Royal Society in the UK, and other European partners, all agreeing to get together to talk about responsible use. So the point of showing you this is not for you to read this, but there was a release after 3 days of, as it says here, thoughtful discussion of these issues. The organizing committee issued, conclusions about what happens in the laboratory. That's number one, which is, we have lots of mechanisms in place to be responsible in the laboratory. The second was about clinical applications in humans. And that has to do with two levels. One is somatic changes. So think of that as any of us on this Zoom who had a genetic disease, a disease caused by a genetic mutation, like the NASH family. And there were a way to go in and alter the cells that were affected using these techniques to make them operate normally, or to take away the thing that was causing the disease. The difference would be the alteration would die with any of us. We're not making changes in our cells that we pass on to future generations. And those are called somatic cells, the cells that are part of our body, soma, right? And the takeaway here is we have had a mechanism in place for a long time about using different tools to do exactly that. It hasn't been very successful, it doesn't work very well, but we know how to be responsible in its use. The third is where it gets more interesting and controversial, and that is making changes to the cells that we use to reproduce. Those are our germ cells, sperm in men, eggs in women. And if you can make changes at the level of those cells, the changes you would make in the genome of sperm and egg or the embryo would be passed on to the next generation, right? The person who was created with those genetically altered germ cells would have genetic modifications that they would then pass on to all their offspring and so on. So you're making a change in the germ line of the individual, but that means you're making a change in the genetic makeup of all that person's children and future offspring, right? all generations going forward. So much more problematic, certainly worrisome cause of how do we think about that and what's responsible to do in that context. So the takeaway was this number 4. So we need a way to keep talking about this because these techniques are new. We don't have a way, we can't do Asilomar. The equivalent of 1975 Asilomar can't happen. And so we need a way for the group at the bottom there, you see it says US National Academy of Sciences and Medicine, the Royal Society, which is the UK version of that, and the Chinese Academy of Sciences to take the lead in creating an ongoing international forum to discuss the potential clinical uses of these tools in humans. So this has nothing to do with what we do with bacteria or plants or animals. This is only about humans, and promote coordination among nations, all really important for reasons that you probably are already thinking, but are gonna come in a second, okay? I'm gonna take a pause and talk about a different technology, which is, I think, instructive. But before I do that, any questions? It's been pretty straightforward so far. Okay, so I'm gonna talk now about something you may have heard of, but it's a different technology that I'm gonna ask you a question about. So the technology is called MRT, or mitochondrial replacement. I'm gonna show you a cartoon now. That's obviously a cartoon drawing of what MRT would look like. And so bear with me for half a second while I explain this. Starting from the left, those are eggs, human eggs from a woman. The thing that looks kind of like a football, big football with a line through the middle of it, is meant to be the nucleus. That's where the DNA that will be that's in every cell resides. The smaller football things that are blue on the top and purple on the bottom are called mitochondria. Those are the cells that make power, the parts of the cells, the organelles in the cell that make power for the cells. Think of them as a battery pack. They have a little bit of DNA in them as well, not as much as in the nucleus, and it doesn't have the same importance in what the organism will be like, but they matter because if you have a mutation, certain bad mutations in mitochondria, they actually are either incompatible with life or they cause really bad disease. So they're pathogenic. And so for some people, the only way to have a genetically related child, it's really only women, because mitochondria are passed from mothers to their children, and they only are transmitted by eggs, not in sperm. So it's all maternal in its passage. For some women, the only way that they can have a genetically related child is to not have their mitochondria passed on if they don't want to have a child with that disease. I didn't say that super well. But think of the bottom. So this is the woman who will have diseased children because the mitochondria are disease causing, right? So she can't help but pass on disease. The nucleus, this thing, from her disease carrying egg is taken out. When you take it out physically, it's literally a pipette is stuck in there and it's sucked out. The nucleus is sucked out with a little bit of the cytoplasm, which is the part of the cell that has the mitochondria. That's why there's a little purple thing going along here with a nucleus. You don't want it, but you sort of can't avoid taking it away. Up here, you've got a healthy egg donated by a different woman, a second woman. You don't want her nucleus, but you want her mitochondria. So the nucleus from that egg is removed similarly and discarded. So he says here, thrown away. So you've got, think of it as a chicken egg. This thing up here is the chicken egg without the yolk, just the white, the albumin, right? And this is the yellow from this egg that is going to be then inserted into this egg. That's what this third thing over here is. So you see the nucleus from this egg is put into the cytoplasm of this egg. And then it's fertilized, that's a sperm being injected into that reconstituted egg. And you can then implant that fertilized egg, this thing here, into this woman's body, and she can have a baby. So her nucleus, a donor cytoplasm and mitochondria put together to make a new egg fertilized by the donor or her partner, and then that's implanted and you get a baby. complicated, but believe it or not, doable. It's kind of like a very a fancy and much more complicated version of IVF. This part over here, number 4, is IVF. This is called ICSI, if you know that from your own lives, intracytoplasmic sperm injection, one sperm injected into one egg to create an IVF embryo. The difference is that that egg was made from 2 eggs. One egg made from 2, right? Combination of 2 put together into one, fertilized by one sperm, and you get an embryo. So all done physically. None of this is done like, you know, it's all with pipettes and sucking out and sticking in, and it's all with magnification in petri dishes in laboratories. So you know this acronym because I mentioned it already, mitochondrial replacement techniques. This is human genome editing. Sorry, I didn't introduce the acronym before. So there's some features that they share. And I'm showing you the covers of 2 reports. That's the MRT report. And then there will be one from the HGE report coming later. So we worry about genetic modification of germ cells and modifications of the germline. So we would say heritable genetic modification for these general reasons. And we probably could identify more than these. One is it safe? Like, it's bad enough to introduce a change that will show up in an individual. It's in some sense more problematic. I don't want to say worse, more problematic if we introduce that in a way that gets passed on to all future generations. It's less concerning if it stays with the individual, right? They don't pass it on. So safety, is it safe? Is it doing what we think it will do? Which is not exactly an ethics concern, but it's obviously about risk and benefit. We need to know that it's safe enough to go forward, which is, you know, the risk benefit balance has to be appropriate, is an ethics consideration. Then there are more sort of clear, frank ethics questions. So sometimes here, like, it's just wrong to intervene this way because it's interfering with nature, or sometimes the shorthand for that is playing God. Like, it's not for humans to do this. If God wanted us to have this power, we would have had it. And it's inappropriate. And that's a sort of hard one for a lot of people, partly because when you start invoking God, then you're invoking religion, and not all religions think the same about use of technologies like this. Some actually think it's important to use them, and others think that we should never use them. So it doesn't make sense to call it all one kind of concern. There's the obvious concern about, well, if we start saying people who have, you know, the diseases that this technique would address, need to have it addressed, then that means they must not be worth living, right? There's some kind of eugenic conclusion that leads us to, and that people who are born with mitochondrial DNA diseases somehow, you know, are worth less than anybody else, and it alters or carries a sort of stigma and inappropriate attitude about those with disability more generally, which is actually, I think, a very fair concern. And you can, you know, see that played out when we don't even have to talk about these technologies, with all of the debate around prenatal testing and Down syndrome, for instance. And then the point I was making earlier, which is there's a different ethics problem when we introduce techniques or use techniques that cross the germline, that alter the germline, such that not only the individual is affected, but the individual's future offspring are also affected. What's interesting about that is we use that, and that term has been used as in the arguments about introduction of technology and the policies that should govern them, as a kind of shorthand everybody understands. It's wrong to cross the germline. But we haven't actually done a good job of articulating why it's wrong from an ethics perspective to alter individuals in ways that will also then be passed on to their future generations. Like, what's wrong with that? And I think increasingly we're suggesting maybe it isn't wrong, especially if we can control the way these technologies are used, such that the things we introduce are actually good, like we've stopped a disease from being in a family, or passed on things that are about health and benefit rather than worrying about disasters. So we have to know that it's safe and will work the way we want. But then a lot of these other concerns are starting to feel like they're less concerning, or they're melting away a bit. All right, so there's the report for the HGE discussion. So the MRT report came out from the same National Academies of Science, Engineering, and Medicine as did this one. The MRT report was issued in 2016, the one you're looking at now in 2017. So, not yesterday, but obviously quite recent, and all trying to foresee the potential uses of these technologies in humans. So there's much more work around the clinical introduction of the use of mitochondrial replacement, because the FDA was getting applications to actually start a clinical trial, so research to actually do this in human embryos in the United States. And so the report that I showed you recommended that certain conditions had to be in place for that to start. And those are listed here. I don't really want to drone on about these because it will use up too much time, but you'll see they're fairly detailed. We need to know that it's safe, that the risk is minimized, especially because we're talking about future children. So among the things that are challenging about clinical investigation, clinical research, when you're using a technique to create a new person, is we can't rely on informed consent. We can't say, well, let's tell people what we understand the risks and potential benefits to be, that we think that they're in balance, and let them decide whether it's appropriate for them to participate. You can do that when we're asking, one of you or me to participate in a new drug trial, if I want to be in a vaccine trial for COVID-19. But you can't ask the person who doesn't exist yet whether they want to be created this way. It doesn't work. And so we've got real challenges to how we set up research in this context, right? We need to know that it's safe. We need to know that it works. That's efficacy. We need to limit it to women who really have no other option to have genetically related children. So their only other option would be to adopt, have a donated egg, which would not be their nuclear DNA, or they just don't have children. Which you may say, fine, why is that such a bad thing? But there's a sort of, some individuals feel it's a compelling need to have genetically related children, and they want to pursue that. And so this would be, available to them potentially. And this is the most controversial thing, so I should say something about it. The recommendation was the way to make sure that you don't have a heritable genetic change, so we don't have a germline modification, is to say, well, just make sure the only embryos you implant are male embryos, because as I said, mitochondria are only passed by women. So if you say only male embryos will be implanted and only males will be born, they can't pass on that genetic modification, because the woman with whom they would be having children would be the donor or the contributor of the mitochondria in that case. So you can stop it very easily, but that's sex selection, which is controversial in its own way. Okay, so Liz, somebody said, isn't that 3-parent baby? Yeah, so this is the way it was reported, and I apologize, I don't know why that got washed out, but this was the screenshot from the announcement in September of 2016 that the first child had been born. Interestingly, the child was born in Jordan, the country of Jordan, Amman, created by a reproductive medicine physician from New York City at a clinic that he opened in Guadalajara, Mexico. Why that? Because it had, by this point, been prohibited in the United States by congressional mandate as part of a budget bill in late 2015. And so he set up a clinic outside the United States and got a couple that didn't come from the US or Mexico to come and be treated, have their embryo created and implanted, and then they went back to Jordan, sort of out of the reach of any of the countries involved. So why did he do that? I think it's on here. He says there are no rules in Mexico, and to save lives is the ethical thing to do. So that's why I went and did it outside the reach of the country where I live or where I practice. The FDA was not happy about this and told them cease and desist, or we're going to strip you of your license to practice medicine in the United States. again, don't read this except to realize that when we started to talk about the use of these techniques to make human embryos or to modify human embryos, so sort of along the lines of what happened with MRT, we were worried might happen with human embryos, as it did, and I'm sure you know it did. This committee, which I actually served on, issued first principles for global governance, right? So we said, here are the, whatever number that is, 7 principles that ought to lead countries to decide what rules to create to govern this technology in their countries. And those are very general. There's a little more flesh on the bones than you see from those bullet points, of course, but it was sort of like, these are the curbs on the road. You decide what the rules are for your particular country within these curbs. That's what we suggested. And then all this other stuff was recommendations related to specifics. And we said, before going forward with human trials to make an embryo using these genome editing tools so that you would have true genetically modified human embryos, you need to have all these 10 items checked off. No reasonable alternatives. Restrict to prevent a serious disease or condition. And you can read through all of these at your leisure, but this is a long list, and it's impossible, frankly, to satisfy at the moment. We don't have, for instance, reliable oversight mechanisms to present uses other than preventing a serious disease or condition. Lots of things would have to be set up between the science has to be ready to the governance has to be ready. None of that was in place, and realize I said this report was issued in 2017. It was issued on Valentine's Day, 2017, February 2017. So we thought, okay, we've now announced to the world what it will take to go forward. This is a locked door, you know, with a padlock and a key lock, and it's going to be really hard to get through. You can knock on the door, but it's going to take a lot of steps before you can actually pass through to do this in humans. We felt pretty good about it. Some things that were true about that report is you can make recommendations about all the principles you want. It's not the same thing as governance. Governance is about law and regulation and policy. And that's not, you know, we couldn't say the policy for even our own country could say this. And I should also say that this was a committee made up of truly international members. So from all across the globe, every continent and the countries you would think would be appropriate were part of this committee, and it's a consensus document. So you can make recommendations about principles all you like, but that's not governance. And translating principles into governance requires saying, well, yeah, those principles are important, but how do you think about which ones are most important? And so, for instance, in the US, we care a lot more about individual liberty and control, autonomous decision-making by us as individuals, and certainly by individuals who are having children, than in other countries of the world. And not to pick on anyone in particular, but China has a very different attitude towards individual liberty than does the US. And so you can say a principle of individual decision-making is important. Okay, yeah, it's important, but important in a different way in different societies, appropriately. I'm not saying one is better than the other, but we need to sort of acknowledge that. And there'll be a different weight afforded to harms, how we think about harm. So in the US, we think of harms really as harms to individuals, not to our culture, which is where other countries and societies come down. So again, harms will be understood very differently, which will also affect the way governance gets cashed out. And then you sort of say, and of course, whatever, you know, one country says, a different country is going to have somewhat different rules in the end, and people will go to the country that they think will serve them best. That's why, you know, someone went to Guadalajara, Mexico, instead of staying in Jordan. Both they could get the thing they wanted there, and the rules were not as strict as they were, say, in the United States. So people will go, as they do, to parts of the world where they can get the things that they want or need. Okay, so now this is all run up to what I think you all know happened. So there was a second international summit at the end of 2018, where actually I was there, I was on the program, And we all thought, gosh, we had another of these things in December of 15. Why are we doing another one so soon? There doesn't seem to be much happening. Okay, it's nice to go to Hong Kong, but it was literally Thanksgiving weekend. Seemed like a good idea at the time, but my wife and I ended up going to Hong Kong together, and we had to like rush out of the door after Thanksgiving meal to get to the airport to fly to Hong Kong, which seemed like really stupid, except that when we got there, there was an announcement that this Dr. J.K. He had created many weeks before and months before a genetically altered human embryo, implanted it into a woman. It turns out more than one women had this done, and in fact twin babies had been born from the technique. So it went from being a kind of, why are we here, what do we need to talk about, to this being kind of this crazy media frenzy and firestorm of controversy about what had happened. And so all of the careful work that had been done, starting with the International Summit in 2015, and then the reports that I showed you, laying out all this careful international expert opinion, somebody who was part of that community, making it actually all the worse, somebody who was known to the people who are working with human embryos using CRISPR and other techniques, but all in the laboratory, and everybody understanding that we aren't going to do this in humans anytime soon. And JK showed up at these meetings and talked about his work as if it was all in the laboratory and prospective, when in fact he was taking the embryos he had been creating and implanting them into women's bodies. And so the program was hastily altered. A special session was carved out for him to make a presentation. The first and only, turns out, time that he showed the slides of his work were at that meeting. We all sat there with our smartphones out and took pictures of every slide as it was presented. He was unaccounted for up until the moment he walked on the stage to make this presentation. He was then grilled by two of the other people who were at the meeting that you see in the bottom left there. And then he disappeared immediately after and had not seen from since, and has been jailed in China so far as anybody knows. So problematic in a lot of ways, which we can talk about, but most sort of worrisome from the perspective of people who've been involved in the governance and ethics and policy debate was, we did all this work, which we thought was pretty good work and careful work and clear work, and then somebody from the same community ignored it and did exactly the thing we were all worried about. So it also damaging very much the notion that science can be done responsibly, even when it's controversial. And so lots of repercussions, as you know. The world community came out very strongly against what Dr. He did. The Chinese government came out very strongly. We can talk about that if you'd like, eventually came out very strongly about what he did, not immediately, but eventually. And we all thought it had been sort of tamped down and we were not going to see any problems with this. And then a Russian geneticist announced that he was taking the recipe, more or less, the technique that Dr. He had pioneered, in his words, and was willing to use it in Moscow for anybody who wanted to come and had a particular mutation that they wanted to introduce, a change they wanted to introduce into an embryo that they would then implant into a woman's body. So very problematic, like a fire, broke out over in Russia. Again, to a lot of recriminations. The Russian community announced that they didn't think it was appropriate, and the Russian government more or less said, you will not do that. We don't want to be embarrassed by the likes of you, more or less. And so it so far has not happened, so far as anybody knows. Where we are now is this committee, the World Health Organization, which you're hearing a lot about now, not in this context, but in the context of the COVID outbreak. But they stood up a new committee, and you see the dates here. First meet on 18-19 March of last year, 2019, and will do its work for the coming 12 to 18 months, which would create global standards for governance and oversight. Sounds a lot like what that other committee did, but with more, they are saying with more specificity, as a way of trying to make sure that Dr. He's work does not happen ever again. And then a second committee, which I am on, this one, was impaneled to work kind of in parallel, very specifically focusing on a translational pathway, which has particular meaning in the world of science. So what steps would you need to take from a scientific perspective in order to go forward with the genetic modification of human embryos with the idea of eventually implanting one into a woman's body? So the same ideas for the mitochondrial replacement technique, but applied to genome editing of human embryos. We've been working for a little bit less time than the WHO committee, and we'll issue our report in middle summer. We're in an interesting moment. Everybody thought we needed to do this super fast because if J.K. Hay could do it, then it was doable by other people in other parts of the world. And there's a J.K. Hay. Lots of people knew him. I had met him in passing. He is not a charlatan, but he's a rogue. And the concern is, how do we prevent other rogues from doing things like he had done? The Russian geneticist would be another example. And we were running really fast, as fast as we could, to issue some strong, good, helpful guidance. And then the COVID-19 pandemic arrived, and we all turned and started to focus on that. But it will come back, right? We'll be through the pandemic at some point, hopefully sooner rather than later, and we'll need to have good answers to these questions. So I'm happy to talk about any of that or anything I didn't talk about. What you saw in the Netflix documentary, if you want, but the possibilities are sort of endless for these techniques. And the focus has been on human applications and very specific human applications, because they seem somehow the most worrisome, but they're by far the furthest off in terms of their ready for prime time application, and sort of how we manage that in the meantime, and all the other techniques that other applications, pardon me, of the techniques in the meantime are sort of what the challenge is before us. So I'll stop. But I should say thank you, really thank you